Welcome everybody, and welcome all our laureates. Please join me on the stage. Thanks everybody. I hope everybody enjoyed the video and with the introduction to another fantastic set of Gairdner Laureates for 2022. It's really great to see everybody here. I've managed to get all the laureates in one room, one Zoom room, for a short period of time. So this is absolutely fantastic. And I'd like to begin by just congratulating everybody once more. All of the awardees joining us today. Fantastic work. Fantastic to see you become essentially part of what we usually call the Gairdner family. You do know that once you become a Gairdner laureate, you're always part of the Gairdner family. It's a very prestigious group of past and present awardees, to be sure. And thanks to all of you, for I know you're all extremely busy, for finding a bit of time today to join us for this Zoom call. And it's a chance for all of you to meet and congratulate each other as well. I know some of you know each other, but this is really your first chance to meet as a group. You will have more opportunities to meet in person. We sincerely hope in person this year in October for Gairdner Science Week here in Toronto, where we'll have you will travel across the country to visit universities. We'll have the laureate lectures here in Toronto and the Gairdner Gala. So we're all looking forward to that. And we are inviting back those laureates who, for the last 2 years, have only been able to celebrate virtually. So I think it's going to be a great occasion. Thanks everybody for joining us from the video to the chat room. You have the opportunity to ask questions for any of the laureates. If you have questions, please put them in the Q&A. If you just want to send congratulations, the chat is sitting there ready for you to do exactly that. So please add your congratulations to the chat and your questions to the Q&A. Meantime, since I have the stage and I haven't seen any specific questions yet, I want to talk to each of you in turn and ask you a little bit about your journey to the prize. Now, all of you actually have always been working on a very particular problem at hand for many years, and you've faced setbacks and detours along the way. And I thought that's what we should perhaps talk about a little bit today. Not, the straightforward idea that you just carry on research and it all falls into place and then there's a great prize at the end of it. We know that science doesn't work that way. So what I want to talk to you and ask you a little bit about what have been the setbacks and detours that have come your way, and also what kept you going when things weren't going quite the way you expected. So Stu, I'm going to start with you. Now, I'm assuming as a practicing hematologist, you always saw the impact of diseases like sickle cell and beta thalassemia. So what kept you working on this problem of globin gene expression over the years? And at what point did you feel maybe it would work and you would actually be able to bring it all together to move towards a potential cure? What was the biggest setbacks along the way?
Well, thank you. I'm really pleased to be part of this fantastic family. I guess I like to solve puzzles. If my wife were next to me, she would say, well, I'm an inveterate crossword puzzle solver, and I'll take any puzzle and try to tease it apart. And really for many years, those of us in the globin field have been interested in how does this switch take place between fetal and adult. And when we started, I think we were very naive about what it might take to actually understand it. There was a period of time for maybe 10, 15 years where I personally thought it was not a problem that could be solved with the technology that we had. And so I guess I'd say that I'm stubborn, I'm persistent, and I like to see things to a final solution of the puzzle. That's partly what kept me going. And I think it's very important really for the patients with these disorders that they need persistence in the laboratory to solve the problem. I think when the press covers discoveries and breakthroughs in treatment, you see the tip of the iceberg. You don't see the part of the iceberg below, which is really the part that makes the difference. And that's, I think, what Gairdner's supporting today.
Thank you. Yes, I think we certainly did. And certainly just looking at the whole story from beginning to end, Stu, it's a wonderful story. And it is a great, it's not there yet, but boy, we're so much closer than before. It's fantastic. So John, same thing. I mean, you've been working on how blood cells are made from stem cells for many years. You're sitting here in Toronto, inspired by Till and McCullough, previous Gairdner awardees, I would point out. But tell us how stem cell background really enabled you to think about leukemia in a different way. And what was your biggest setbacks along the way? Oh, you're mute.
You know, done this for 2 years and you still end up yakking and move on. You know, I think I'll start off with the failure first, or the setback first. It's interesting because it actually has a track back to Stuart, and it'll perhaps jog a memory of his. So, you know, we had done this work of trying to figure out how to test for stem cells, which was, you know, you can only test a stem cell by watching what it does, and what it does is regrow a blood system. It's hard to do experiments in people. Nobody wants to volunteer for my experiments. And we figured out this way where we could put human cells into mice that are immune deficient, and they would grow. And so we now had a way to study human stem cells, albeit in the context of a mouse. And the key work I did as a postdoc with my amazing mentors, Alan Bernstein and Bob Phillips, was to put genes into stem cells. This is in the beginning of gene therapy. It was going to be the revolution in the 80s, and so we were doing that. And so we tried to put genes into human stem cells. It worked beautifully in the mouse. We could put it into human progenitor cells, which are another cell sort of in the bloodstream, but we could always fail to put genes into the cell that engrafted a mouse. And the trainee working on this did it again and again, and they said there must be a problem with this again, until we finally stepped back and thought about our data, and we realized that what that really meant was that the way to put genes into progenitor cells is very easy. It works beautifully, but we actually had a different cell that wasn't a progenitor cell, because we could never put a gene into it. It still worked. It still regrew blood system in a mouse, but we could never put a gene into it. And this is the beginning of when people were getting to realize that, you know, gene therapy was going to perhaps have some problems. And I think, you know, our work was a small piece in a very important committee that Stu co-chaired that basically brought light to, you know, gene therapy and some of the setbacks. But for us, it was a setback. We couldn't put a gene into the cell, but for us, it was the aha moment that we had a different cell that was different than any other cell in the bloodstream. So, you know, the setback lemons to lemonade, I think is the thing.
Yeah. And again,
if you want me to answer the first part of your question, but I answered that one wrong.
That's fine. But again, persistence and sticking with it, and learning from different systems too. So a lot of crosstalk between research from different people, right? And then that's another message, that nobody does this alone. And obviously it's your team of researchers, but also, you know, things you learn from other people in the field. So it's always an interactive, collaborative process. Okay, so I'm going to turn to Zulfi now, which is, we're moving in a totally different direction from the sort of, you know, molecular biology of stem cells and blood progenitors into really trying to have an impact on maternal and child health globally. So Zulfi, what has been the driver that keeps you going, and some of your biggest setbacks along the way?
So Janet, what kept me going was the hope that we could address some of the issues that got me started in global health in the first place. As many people know, I'm a card-carrying neonatologist. So for the first phase of my career, happily worked in a newborn intensive care unit trying to set up services in the large city of Karachi, and growing increasingly frustrated by the year to see how little impact just working in a hospital had on the larger issue of disparities and difficulties in reaching people in rural populations. So my stepping out of my comfort zone into trying to do something with communities, community health workers, outreach programs in rural populations was a leap in the dark. And many a setback in that early phase was largely related to just exploring new grounds. I mean, just trying to do things which hadn't been done before. I would say over the years, we've been rewarded enormously by the success we've had, but also have learned tremendously from the failures. So not every cluster randomized trial amongst the dozens that I've been involved in has been a success. But I would say with great confidence that we've learned a lot more from the negative studies than we have from the positive ones. And learning what doesn't work in which particular circumstance has been particularly, I would say, salutary and important for global policy. My one regret over time, as I've again mentioned to people, is that when you get involved in research and work of this nature that addresses not only abject poverty, but some of the social determinants of poverty and ill health, there is just so much out there that you wish that you had reached out and done something additional, a little more. And in science, you can't do everything. So as I have built a portfolio of global evidence around interventions, I also realized what I have not worked on. And I'm in some ways at this stage of my career, turning to things that I wish that I had turned my attention to decades ago. Things like environment, things like climate change, some of the fundamental issues around food security. But nevertheless, it's been a fascinating journey, Janet. And as you know, it's not quite over, and I'm still enjoying learning as I go along. Thank you.
That's great. Thanks very much, Sophie. I think that question, of always that you learn from the negatives as well as the positives. And I think that's something that, again, is a good message for people who don't really know how the scientific process works, that we learn perhaps even more from what doesn't work. And we have to acknowledge negative results, whether it's clinical trials or whether it's a molecular study.
Absolutely, absolutely.
It's fundamental to the process. So Deborah, I think what I just said also applies obviously to some of your trials as well. You learn when you have clinical trials in critical care research, some of them are going to say, yes, this is right. And some of them are going to say, you know what, what we said we thought was the best practice is not the best practice. You have to do that. So, but you're an intensive care physician. You're incredibly busy. Your days are packed, especially right now with COVID. And I know how hard you work and how hard it is to get a hold of you because you're so much committed to your patients. But you made time in your career to develop clinical trials to improve the standard of care. And you also made time to develop programs to provide compassionate end-of-life care. So what made you so committed to this evidence-based medicine? And again, biggest setbacks along the way?
Thank you, Dr. Hassan, and everybody for this huge honor. On behalf of Canadian Critical Care, I offer a few reflections. When I began my career many years ago, looking after the sickest patients in the hospital, most of our understanding of what was going on was based on pathophysiology, often animal data. We did journal clubs based on laboratory studies and observations that didn't have that much to do with critically ill patients. So when we began in Canada, we knew we had to study patients, not a single problem, not a single organ, not a single disease, but critical illness is very broad. And in its entirety, it involves basic and advanced life supports. And the challenges were myriad. Patients come in with one problem and develop other problems. So we decided across the country just to band forces, recognize we could achieve more together than we can as individuals. And the vast expanse of our country sort of met with collective enthusiasm for working together. And as you pointed out early, this collaborative spirit is key because there's so many bumps in the road. Some days it feels like we're all climbing a mountain. And while we may learn more from the challenges and setbacks than our successes, I think we do need a long view and a clear view. And something important in science, I believe in acknowledging explicitly, is the role of humility, because that can help us understand when we need to step back, maybe stop, take another look, retrain, try to work with new collaborators, and maintaining a humble and an open stance, I think has helped Canada set the stage for large clinical trials, which have really come into play in the pandemic, as you pointed out.
Great, thank you. I think those are good messages about humility, because the challenges are always going to be perhaps bigger than we know. So let me move on to Kathleen and Drew. I'm going to ask you the same sort of question, but you can do it in turn. I think many of us have heard some of your story already, which is a fantastic story of really persistence against the odds in bringing these messenger RNA vaccines through to the clinic. It's an absolutely amazing story. I'm going to ask Kathleen, not so much the setbacks, because we know the setbacks. When was the turnaround moment when you finally saw that maybe your commitment over all these years was going to pay off? And then Drew, what was it about Kathleen and her research that inspired you to partner with her and make this partnership that's been so successful? So Kathleen, when did things turn around? You're mute too.
As our fellow overleas are saying, that they all believed in something that maybe around them people did not believe, and it was maybe undoable. And so definitely that the question was about, the RNA, why it is causing inflammation, and realizing that we realized that why it is inflammatory. And the next question was whether we can make it non-inflammatory, still coding for protein. And so when we could do that, of course, we realized that what we set out, it is doable. But as all of our colleagues here today, that we just, all of us believed in something, and that was the drive behind it, that we pursue and were, not giving up despite all of the failure or difficulties, what every scientist face constantly difficulties, and then they just somehow, push themselves through. And so that was the reason, the realizing that the modification is important, but at that point we didn't know that whether we can make messenger RNA such, which would code for a protein. And then because nobody have done modified RNA, nucleoside modified, and being able to do it, and this was translating to a protein. And that was, really, we knew that this would be great. It is just, other people at that point, they did not. But we were not disappointed that much, because there were so many other things we had to do, purify that RNA and so on. And so we just focused on those other things.
So you just kept going a bit at a time until the last puzzle, the part of the puzzle. We've heard that as well. It's a puzzle that you have to sort out all the pieces that you go through. So again, a common story. It's a great story. And Drew, so what drew Drew to Kathleen? How did this happen, and why did you really feel that her research was going to make a difference?
I think what drew me to Kathleen is just my interest in research. And, I collaborate with hundreds of researchers across the world. I never say no to anybody who wants to collaborate and investigate, because my role in this process is to learn, is to investigate, is to try and understand the future knowledge, to continue to understand, and then to take that and to develop it into new therapeutics. So, I ran into Katie at the photocopier, and she worked in RNA. I was interested in making a vaccine, and RNA was a great route to take. And that's what started us working together. I think what's more important is what continued us working together is that we both viewed research as a way of learning, and we were both completely open-minded, and were interested in understanding what the results meant, not expecting results, but just doing an experiment, seeing the results, and trying to understand what that meant. And we were both fixated on that, and that's why we kept working together for 25 years to try and understand the research and try to understand what RNA did, how to make it better, how to make it into therapeutics, and we both continue to do that.
That's again, I think, common stories across everybody here. They're just following the story, doing the research, being humble, not trying to think you know all the answers, but doing the research to find out. Fantastic. And so Peter, I haven't forgotten you there. Another story of dedication to a research area, lipid biology and its applications over many decades. Fundamental science leading to lipid nanoparticles. You were always, I think, looking for applications and spinning off companies to move things forward. What were your biggest setbacks and successes along the way, Peter?
Yeah, as you can probably tell, I'm in Heathrow Airport at the moment, and so there's if there's a little bit of background noise or a lot of it, you'll understand. Yeah, it's just a couple of words on what drives some of the work that we do. There's really two. If you want to think about two rather ridiculous things in medicine. One is that we take drugs like anti-cancer drugs, and they go everywhere in your body, and only 0.001% actually gets to a tumor. This obviously requires a more accurate delivery to the site of disease. And the other one is that we use small molecules because those are the only molecules that can get inside cells to some target protein. And so that means we can't use the bigger molecules, such as the nucleic acid-based drugs, as direct therapeutics. And so that's really those are the drivers for me with the drug delivery approach. But I think this is going to revolutionize medicine if we can use the larger molecules, and obviously the mRNA is a great example of that. And eventually we'll be able to also deliver cancer drugs, I think, much more specifically to where they're needed, and avoid some of the nasty side effects and dose-limiting side effects that are there. So those a third area is that again is pivotal for me is the aspects of teams. Research is really a team endeavor. And so what I've enjoyed most in my research career has been being part of teams where you have a lot of fun. You discover a few things. I can see that John has been celebrating a few things, like his backdrop of many champagne bottles. So these are it's that part of the celebration of doing science that is particularly attractive. It's a wonderful way to spend your life, that's for sure.
So yeah, I think everybody would agree that that's perhaps something we didn't say, that we do have fun as scientists, and that's one of the things that keeps us all going. So I've got some questions from the Q&A that I'm going to throw out. Perhaps one of them mostly to the messenger RNA vaccine people, because it's come up most relevantly there. But the question is, we've seen from David Rose, we've seen the gap that exists between your amazing achievements and the lack of acceptance of some portions of the public for emerging therapeutic strategies. In this case would be vaccines. So what is our role as scientists, and how do you approach this issue? So Kathleen, Drew, you want to go?
I spend a lot of time on nights and weekends talking to lay audiences. I'm a physician researcher. So I do see patients, and I spend a lot of time trying to explain to them why the RNA vaccine is safe and why it's so effective and why they need to take it. And 20 years ago, a couple % of the population refused to take vaccines, and we knew why, and we knew that there was nothing we could do to convince them. I think times have changed. Social media has made things much worse. And that's made vaccine acceptance much more questionable. And I don't really know how to address this. And that's one of the most frustrating things about my life right now is I wish I could snap my fingers and have everybody take their vaccine. That's not working. And I don't know how to solve this problem. And I could use as much help from as many people around to try and figure out how do we make vaccines acceptable? How do we make science acceptable? And I think that's the big problem right now.
Anyone else want to comment? Zulfi, on a global scale, do you have any comments on this?
On the global scale, Janet, there is the whole issue of vaccine inequity, which is around supply. We don't have as much hesitancy as you tend to see in well-endowed circumstances. My biggest challenge as a global health researcher with COVID vaccines is the enormous vaccine inequity that we still face. Less than 10% of low-income countries have received any form of vaccines. And if they have received any, they typically are not the best vaccines, such as the mRNA vaccines. They typically happen to be the other types, inactivated and some of the vector-containing vaccines. So the big challenge that we have is not just in having a product available and having the advocacy around supporting it, is to how we as a global community can work towards vaccine equity in delivery, in making sure that this becomes available for a truly global problem. But I want to add a note of optimism here, because this was exactly the situation that we had with childhood vaccines some decades ago. I keep telling people that, at the time when my own children were growing up, vaccines were not available to many children in low and middle income countries. I remember when one of my sons was born, that I actually bought the vaccine in North America to take back home because it was just not available for the hip conjugate vaccine. Today, those vaccines, Janet and colleagues, are available to every poor child in the world. And in many countries of Africa and Asia, we have over 90% vaccine coverage. So we've been able to do it through a concerted mechanism and through an innovative mechanism of financing. I think with COVID, it's early days. And despite COVAX, I think we haven't really explored the opportunities and mechanisms for making some of these fruits of research and things that are principally evidence-informed global responses available to each and every person where they need them. That's our big challenge right now.
Yeah, and a sort of follow-up, not on vaccines, but I'm going to turn this one to Stu. And I was about to ask the question that Mary Claire King has just put in the Q&A. And she says, could you speculate on health policy steps that would be necessary to enable gene therapy for hematological disorders like sickle cell disease to be used by a large number of patients globally? So it's another issue of access to these kind of therapies. Stu?
Yes, well, I think John could probably comment on this too, but I think that access may be, in this case, a question of economics, because the therapies are likely to be or will be expensive. I think that if one does economic analysis, though, one will eventually save money to the health system if one has an early gene therapy as opposed to treating, for example, an individual with sickle cell disease throughout an entire lifetime. I think the key, though, to expanding gene therapy is going to make it more an easier procedure, a less expensive procedure, easier procedure. Currently, the practice is to take stem cells that are mobilized and treat them in the laboratory, modify them and reintroduce them into the patient. That's cumbersome. I think it needs to be done as either an in vivo therapy of some sort or some sort of much easier approach. And perhaps the lipid nanoparticle approach is one way to get there with gene editing components. I still think there's a question about efficiency because particularly for the globin disorders, the correction has to be quite efficient in terms of the number of stem cells that are modified. But, I think that is at least conceivable going forward. I think that should be the direction. I think there are people working on that for sure.
Okay, I'm going to turn to another question from the audience. This is from Frank Gairdner, whose name tells you that he's part of the Gairdner family. He's one of our members of our board of directors. And this is for anyone to answer. I'll throw it out. He's curious to know, what are the biggest impediments to research and curiosity or discovery research in all the different fields. And what can be done to alleviate those? So what are the barriers to really ensuring that we continue to invest and allow broad-based discovery research? Who would like to take that one?
I can start with, because some of this is actually within us, the scientists. And what I have seen, the young one, is sometimes, that, they when they do research, somehow they want to please the supervisor or working for, the company or the university, instead of just focusing of solving, problem. Because, if you try to please the boss, you are set up for disappointment. And what other young, I ask them and, experience with this, they feel so intimidated sometimes, to going, and everybody knows everything. And then they kind of get scared, or others are looking at, and comparing themselves to other fellow scientists who might work less and get more paid. And then they get dissatisfaction instead of just focusing what they can do on their work. And so some of them is within, the scientists themselves can change that. And maybe others suggest for the system.
So maybe I can jump in, Janet, and say, I typically tell my students and staff that one of the biggest challenges you face in global health at times are the 3 different forms of poverty. And they also reflect on students, trainees. So first is the poverty of income and assets that we all know, just being poor and not having enough resources as researchers in a different place. But the other 2 forms of poverty are things that people don't talk about, but they are very important. And overcoming those and moving beyond them is a fundamental underpinning of some of the work in global health. And that's the poverty of hope and the poverty of imagination. To do the kind of work that many people do in global health, you have to be an optimist. You have to have the faith in the inner workings of science that can help alleviate some of the misery, poverty, disparities in the world. If you did not believe in that, if we did not know that some of those interventions can make a difference, we would not be in the business that we are. So I have seen in my own lifetime, over the last 25 years, child mortality rates reduced from 10 million deaths in children under 5 every year to less than 5 million deaths as we stand today. That is the fastest reduction of child deaths in the history of mankind. And it happened in the Millennium Development Goals. Principally why? Because the world got together not only against a global compact, but worked together to put evidence-informed interventions into policy and practice. So you have to have that hope, and you have to have that imagination to think laterally. A bit like Catalin never gave up on the idea that mRNA as a process for giving vaccines would work. So important for young students not to give up hope.
So that takes us really, I think, probably to the last question we're going to have time for, which some of you have already, and Zulfi, you've just really expanded on, but I'm going to make sure everybody gets a chance to answer this one. And it is, of course, what advice would you give to early career scientists starting their careers now? What's your best advice for them going forward? And who am I going to turn to? Deborah.
Thank you. It's a wonderful question. There's so many comments. I know we probably all have, could talk for ages. One of the most important things I think young folks should consider is the importance of working with individuals who don't think in the same way as you do. So we avoid having a group think. We make sure we work with and contact with and discuss with people who have different points of view. That helps us be sure we don't oversell our own. And it underscores the key role of teams and collaboratives that I mentioned we've got in Canada with the Canadian Critical Care Trials Group that I've been so proud to co-found. So I think those are key messages over and above finding and fueling your passion, making sure you keep an open stance and talk to and listen to. We tend to listen less than we talk as scientists. Keeping an open mind to new ideas, new ways of looking at things, and paralleling that with the dogged determination that we've all, I'm sure, enabled over the years.
Peter from Heathrow.
Yeah, no, I think we're just entering right now an extraordinarily exciting area in therapeutics as the potential of gene therapies comes into play. When you're obviously using lipid nanoparticles or potentially other delivery systems. But this is remarkable because all of the molecular biology that we built, that knowledge we built up over the last 50, 60, well, 70 years can now be used in a very direct way therapeutically. And so this is, I say for younger people, this is an enormous opportunity. This will be the third generation of pharmaceuticals that are being developed now. The first generation being small molecule drugs, second generation biologics. And now we have these gene therapies that really, the vast majority of diseases, we can go after them very directly. So I think for students in the life sciences, this is a golden era that's opening up, extraordinarily exciting. And I would say that if I was in my 20s, I'd be doing what I'm doing now, and only more so. So.
Thank you, Peter. Stu, how about you?
I guess I recommend just following something you think is interesting and not necessarily what the mainstream is doing, because if it's interesting, it may become the mainstream years later.
Okay, John, I think I haven't asked you yet.
I think in terms of what my recommendation is that identify a big, what are you interested in, but identify a big question, a long range question. At the end of the day, that can do you for a whole career to sort that out. But I think have a good question, in our case, it's really one that, we were given from Till and McCullough, which was, what makes a stem cell a stem cell? And, 30 years later, we're still really asking that in one way or another. So, find a good question and stick with it.
Find a good question and stick with it. I think that's a message that all of you have shown the importance of doing exactly that, every single one of you. Drew, your advice to a young scientist?
I think it's not so much advice, but I think as senior scientists, we need to look beyond our communities. We need to look worldwide. We need to go to regions of the world where education is limited or where rights are limited, where access is limited. And we need to extend science and the ability of people to get into the science, because there are brilliant scientists all over the world. And we need to figure out how to give them access to getting into research and getting into science. It needs to be a worldwide effort.
I think that's very important. Obviously, you and Zulfi need to get together and have a discussion on this issue, I think. And finally, Kathleen, I think you get the last word on this one.
So very similar things I would say to the young one is, they had to find something what they like. And if they like to be challenged and excited and solving puzzles, as just Stuart said, that they should select science and be excited and doing something they love, and they don't need hobby, then that would be, what they always want to do. So
I think there's so many common messages from all of you. This has been a great discussion. I hope you've enjoyed it. I certainly have. I hope the audience has enjoyed it. Thank you for the questions. Lots of nice messages in the chat for all of you. We'll try and make sure you get all those messages. And so congratulations again to everybody. We're going to have a great year. We will see you in person and we will be in touch. Please, everybody, check out our website, Gairdner.org, and our social to find out what's going on. We have some global perspective panels coming up over the next few months, and then we head up into Science Week in October. So thank you very much, everybody. Thank you very much indeed.
Thank you.
Thank you.
That's great. Yeah.
Bye.